Hello students, myself Dr. Vinita Rathore, Associate Professor Zoology at SMB Government PG College, Nathadwara. Now this video is in continuation with the earlier video of Meiosis Part 1. After completion of prophase first, the next stage of the meiosis is metaphase first. In this metaphase first, the bivalent or the homologous chromosomes arrange themselves in the plane at the equator, forming equatorial or metaphase plate. The centromere of each chromosome is directed towards the opposite poles and the arms of the chromosomes are faced towards the equatorial plate. In this stage, all the three types of the fibers, that is, continuous, chromosomal, and interzonal fibers, are visible in this stage of the cell division. Here in this diagram, we can clearly see that the centriole reaches at the opposite poles of the cell. Spindle fibers from one pole of the cell attach to one chromosome and spindle fibers from the opposite pole attach to the homologous chromosome. After metaphase first, the cell enters into the next stage of meiosis, that is anaphase first. Here, the chromosomes move to the opposite poles. After spindle attachment is complete, the microtubules of the spindle fibers begin to shorten. As they shorten, they break the chiasmata and pull the centromeres towards poles, dragging the chromosomes along with them. Because the microtubules are attached to kinetochores on only one side of each centromere, the individual centromeres are not pulled apart to form two daughter centromeres as they are in mitosis. A difference between mitosis and meiosis is that sister chromatids remain joined after metaphase in meiosis first, whereas in mitosis they separate. During anaphase first, original chromosomes separate. So, reduction in the number of chromosomes from 2n to n number takes place. Yet, the sister chromatids remain together. When the spindle fibers have fully contracted, each pole has a complete haploid set of chromosomes consisting of one member of each homologous pair. At the completion of anaphase first, the cell enters into the telophase first. Here, the homologous chromosomes complete their migration to the different poles because of the shortening of the spindle fibers. The nuclear envelopes organized around two groups of the chromosomes and the nucleolus, which disappeared in the earlier stages of the prophase, they also start appearing, or reappearance of the nucleolus is there. After completion of the telophase first, cytokinesis takes place. Cytokinesis involves the formation of a cleavage or a furrow resulting in the pocketing of the cell into two cells. At the end of telophase first and cytokinesis, two daughter cells are produced. Each is with one half of the number of the chromosomes, or you can say haploid set of replicated chromosome of the original parent cell. After completion of the cytokinesis, the cell undergoes into the next stage, that is called interkinesis. The interkinesis can also be termed as interphase second, and it is very similar to the interphase stage of the cell division. Meiosis second. The second meiotic division is sometimes called as the homotypic division or the equational division, because the chromosome number remains the same in each cell before and after the second cell division. The second meiotic division is of mitotic in nature, and it divides each haploid cell into two daughter haploid cells. This meiosis second consists of four phases, that is prophase second, metaphase second, anaphase second, and telophase second. The first phase of the meiotic second division is the prophase second. During this phase, the chromosomes reappear again and then split longitudinally to produce chromatids. The sister chromatids remain connected with each other by a single centromere. Nucleolus starts disappearing and the nuclear membrane is broken down into pieces and then disappears completely. It is followed by appearance of the spindle fibers. After completion of the prophase second, the cell enters into the next stage, that is the metaphase second. Now, as the breakdown of nuclear membrane completes, the chromosomes arrange themselves in a plane at the equator, forming equatorial plate. Each pair of sister chromatids contains single common centromere. Here, the free arms of the chromatids are directed towards opposite poles and the centromeres towards equatorial plate. After completion of the metaphase second, the cell enters into the next stage, that is anaphase second. In anaphase second, each centromere divides into two, thus every chromatid is provided with its separate centromere. Then the non-sister chromatids move apart towards opposite poles. The movement is checked up by the spindle fibers as usual. After completion of anaphase second, the cell enters into the next stage, that is telophase second. One member of each pair of homologous chromosome present in each pole. Each chromosome here is referred as monad. Monad is a combination of maternal and paternal genetic information. The nuclear membrane reforms around the set of chromosomes at the poles. Following cytokinesis and finally 4 haploid gametes results from a single meiotic event. Functions of meiosis. The main function of the meiosis is production of haploid gametes so that at the time of fertilization, restoration of 2n number of chromosome occurs in normal somatic cells. Second, production of tremendous amount of genetic variation. As we know that during chiasmata formation and crossing over, the genetic material is exchanged and the variation is quite possible. So, with the help of this meiosis, we can find genetic variations. The third function is segregation of the 2 alleles of each gene. This takes place due to pairing between the 2 homologues of each chromosome and their separation at the first anaphase stage. Due to meiosis, recombination between linked genes due to crossing over during Pachytene stage is possible. Meiosis facilitates segregation and independent assortment of the chromosomes and genes. In sexually reproducing species, meiosis is essential for the continuity of generation. Because meiosis results in the formation of male and female gametes and union of such gametes leads to the development of zygotes and thereby formation of new individuals.